Hello, and a very warm welcome to everyone joining us for today's Select Science webinar entitled Advanced Molecular and Cellular Technologies for Better Monitoring of HIV-Positive Patients. My name is Lindsay Forsyth, and I will be moderating today's presentation. I am delighted to introduce today's speaker, Andrea Cossarizza, who is Professor of Pathology and Immunology at the University of Modena. Today he will be discussing advanced single-cell approaches for HIV research and treatment. Just before I hand over to Andrea, I would like to mention a few things. You can ask a question for the Q&A session at any time during the webinar by clicking on the Ask a Question button at the bottom of your screen. You can also view the presentation in full screen by clicking on the tab at the bottom right of the media player. You can also download related information using the link in the Resources tab at the bottom of your screen. The on-demand link will also be provided after the webinar. Without further ado, I would like to hand over to Andrea and thank him for presenting to us today. Please go ahead.
Thank you very much for the kind invitation to present a seminar on the advanced technologies that we can use for improving the monitoring of patients with HIV infection. And I will try to present some data from my lab in the framework of a general overview of what is today the infection with HIV and which are the strategies that people all over the world are trying to pursue in order to fight the virus and to keep the state, the patient, in the best possible status. So which are the learning objectives of my presentation? I will present the latest advances in the research of HIV and especially in the treatment of patients with HIV infection. I will show you the complexity of the viral reservoir and which are the possible therapeutic approach to kill infected cells. I will show you how you can use a very sophisticated and new technologies like cell sorting in a special environment which has been designed to manipulate samples from patients with HIV infection, but not only patients with HIV infection. Actually, all human samples are at risk of an infection, so I will show you how you can put very important and big instruments under a cabinet to work safely with the blood or lymphocyte of cells from patients, for human beings actually. Finally, I will show you what we have done in the last months regarding the quantification of HIV in highly purified, isolated T-cell populations, and how we could not only measure the presence of the virus, but also to analyze the age of the cells by the two different approaches, which is the first of which is the quantification of the T-cell receptor rearrangement excision circles that are called TRECs, and the second approach is to measure the telomere length. Which are the most important facts in the field of HIV infection today? Well, the news is that actually there are 17 million people who can access antiretroviral therapy, and most of them, actually more than 90%, are and have very good results from therapeutic point of view, and they have the viral which is completely suppressed, so you do not find the virus in the plasma anymore. And the bad news is that there are about 40 million people, actually 36.7 is the estimation by UNAIDS. That means between 34 and 39.8 persons are living with HIV all over the world. And we know that every single day 2 million people every year, 2 million people become infected with HIV. More than 1 million people die for AIDS-related illness. And at present, from the beginning of the epidemic, 78 million people became infected with the virus, and 35 million people have died because of AIDS-related illness since the start of epidemics. So which are the key points of the fight against the virus. The first important thing is that it is crucial to stop the spreading of the infection as much as possible. And by the way, tomorrow it will be December the 1st, which is the World Day for the fight against HIV. And what I always say to people and to my students and to everybody I meet in such occasions during some public conferences or meetings is that it is possible to stop the infection with prevention, and prevention remains the key words that we have to use in the fight against the virus. So we have to prevent the spreading of the infection as much as possible. Second, we have to do another thing, which is the try to eradicate the virus from patients, which is not very easy because the virus integrates into the host DNA, and it is really complicated to have the virus expressed and to kill cells that are infected. However, eradication of the disease, of the infection in patients with HIV is still the holy grail that everybody is trying to reach with different strategies. And in fact, for this reason, different strategies are under investigation because we need to know how these strategies can be improved and how the virus reacts to the strategies. I am not going to talk about the different therapeutic strategies, but just to give an idea, we can combine today pharmacological strategies. That means we can use drugs which are very potent and are very able to block the virus, along with compounds that are able to activate the immune system and to kill infected cells. And we have to remind that the strategy that the immune system uses to fight a viral infection like HIV is to kill cells that are infected, which should be a bit nonsense because you kill cells in your body. But the logic that the immune system has is that if a cell has a virus integrated into the genome, the only intelligent thing to do is to kill the infected cell. In this way, the cell is not able to produce virus anymore. So we have to consider this in the framework of all the possible strategies for the control of the infection, because we have to not only to inhibit the production of the virus, but also to enhance the response of the immune system to the virus. I mean, the response to the virus means the presence of cytotoxic T cells that are able to kill infected T helper cells. And I will show you in a moment during my presentation which is the importance of cytotoxic T cells that are usually CD8 positive and are easy to be recognized by flow cytometry or by other systems. So it is clear, however, that we need to design strategies, but we also need to have diagnostic tools, because measuring the presence of HIV in all the possible forms of the virus is crucial, and it is also very important to measure the virus everywhere. Everywhere means that a number of cells in the body can be infected by the virus. I mean, not only CD4 cells, but also cells present in, not only those present in the blood, but also those present in tissues, in the gut, in mucosa, and everywhere. And we know by several papers published in the last years that a number of cells different from CD4 can be infected by the virus. So the infection is really something heavy and present in different parts of the body. For this reason, it is crucial to have the possibility to measure HIV in all the forms that the virus has. Actually, there are several forms that the virus can have when it enters into a cell, and I will show you in a moment what we have done by measuring the total amount of the virus within cells, but we have also technologies to measure the integration of the virus into the DNA of the host, and we have also methodologies for measuring the presence of circular forms of the virus in the cytoplasm. And circular forms are those that give a more precise idea of what is the production of the virus into the cell. It's a sort of intracellular viremia, if you pass the term viremia for what happens into the cell. So it is clear that eradication of the virus from the host requires a number of different strategies that combine together the use of potent drugs and vaccines, the compliance and adherence of the patient. And this is important because well, I remember the old times when we start treating patients with drugs. I'm talking about 30 years ago, even more, when we had only one single drug, which was the zidovudine, able to fight in some way the virus. And when other drugs became available, patients had to take a huge number of pills per day. And of course, if you had to take maybe 8 or 12 pills per day, one every 4 hours or something like that, the adherence is very low, and so it means that it is difficult to maintain for a long time a high plasma concentration of the drug, which is able to block the replication of the virus. So the new strategies and the new drugs are very potent and very easy to use, also because in some cases now there are therapies that you can take once a day. There are treatments, very simple, very easy to use, and everybody who has had problems with blood pressure takes 1 pill or 2 pills per day. Well, today, fortunately, the therapy for HIV infection is similar. I mean, you have treatments that only need 1 pill per day, so it's very easy, it's very simple. And it means that if on the one side treating patients is not a big problem, on the other side, to monitor the immune response and to monitor viral reservoir is indeed a problem. To monitor immune response is complicated for many reasons. One of the main reasons is that what people do usually is the count of CD4 T cells, and the count of CD4 is not dramatically expensive, but can be done in almost all laboratories, and many people can use the CD4 cell count to monitor the immune response. The problem is that CD4 is not one type of cell. CD4 are a huge amount of cells of different types. So it means that within CD4 we have different types of cells that do different things. For example, we have cells that perform that produce cytokines like interferon gamma, which are pro-inflammatory. We have cells that produce interleukin-4, which are allergy, can provoke allergy, that are linked to allergy. We have Th9, we have Th17, which are pro-inflammatory as well. So we have different, and we have, of course, Tregs, I mean, regulatory T cells, which are able to inhibit the immune response and to suppress the response of the immune system. So within CD4 cells, we have a universe, and it is very difficult to monitor, also because usually the laboratories do not perform more than the CD4 count. And so if clinicians want to know in a more detailed way how the immune system of the patient is, it is complicated and it is quite expensive. Similar problems for the viral reservoir. Usually what we clinicians do is to measure the amount of virus present in the plasma. And if you do the plasma viral load, you have an idea of what is the production of the virus, but you do not have an idea of what are the reservoirs. So it means that with the antiretroviral therapy, you can block the production of the virus and you do not find the virus in plasma anymore. But it is not that the virus is no longer present. It is that the virus can be present inside cells and in different manners, in different amounts between different types of cells. So it means that the CD4, which are activated, which are memory cells and so on, are more prone to be infected than naive T cells or CD4 T cells that are not activated. So in different types of CD4 cells, you can have different degrees of viral load, of presence of the virus, and it is very important to measure a reservoir in order to understand whether you have a sort of production of a virus instead of having the block of the production of the virus. So monitoring immune response and viral reservoir is crucial in order to understand how the infection goes on, which are the reservoirs, and which is the best strategy for the eradication in order to have the immune response and immune activation, and to realize how important is immune activation during an immune response, and in order to look for the cure. And this is the outline of my talk, along with a short part on what is feasible and what is the best thing that we can do to measure the infection, to measure the reaction of the patient and in the immune system, and how. And this is what I will try to present in my talk. As I mentioned, potent antiretroviral therapy can reduce viral load to a very low, if not undetectable level. So it means that with the classical methods that have been developed years and years ago, you are not able to measure the virus in the plasma anymore, because the level of the plasma goes below maybe 50 copies of virus per ml, if not 20 copies, if not 2 copies per milliliter. And this is due because of the possibility to use a huge number of compounds, and you see in this slide what was in the market a couple of years ago. Actually, there are new compounds arriving in a short time, and there are drugs of the category of nucleosidic reverse transcriptase inhibitors. There are non-nucleosidic reverse transcriptase inhibitors. There are protease inhibitors. There are fusion inhibitors, entry inhibitors, integrase inhibitors, and combination of different classes. And this is important because, for example, with a compound like Atripla, you can use 1 pill per day on and have the complete control of the viral production. However, the problem is that, as I mentioned, maybe you can, in most cases, up to 90% or even more, you can obtain a complete control of viremia. So it means that you do not find the virus in the plasma, but the virus still remains in cells. And so the problem is that since the antiretroviral therapy can prevent the reinfection, it is nice, it works well, but antiviral therapy is not able to target the reservoir. So it means that the reservoirs still remain intact during antiviral therapy, and the virus still remains in the body, ready to come out in the first possible moment. So it means that if you have a cell which is infected and is not producing virus, if you activate the cell starts to produce virus. So it means that the reservoir can be triggered and the production can come out in any moment. So this is the reason why people are trying to obtain a complete eradication of the infection and to eventually kill cells that are infected, because you cannot get rid of the virus from the DNA anymore. So, and the result is that if you stop antiviral therapy, you have a rebound of the virus production because you reactivate the reservoirs, and for this reason, the therapy has to be taken for life. So it means that patients have to take pills for a number of years, which is fortunately now very, very long, and actually the life expectancy of patients with HIV is very, very similar to that of patients on normal, not normal, of people without the infection, And we have done a study here in Italy, in North Italy, in comparing almost 10,000 patients with HIV with the population living in the north of Italy, which is 16 million people, and we could analyze, together with a group of the infectious disease clinics and the epidemiologists and the National Institute of Health, the life expectancy that was found to be only a couple of years shorter than that in the general population. So it means that life expectancy in Italy is very long. We live more or less 82, 83 years, and the life expectancy of a patient with HIV, if the therapy works and if it is taken from the beginning of the infection, can be as long as 80 or 81 years, which is really something big, I mean. So, but the question is, of course, why do we need to cure HIV? Well, first, because life expectancy remains is reduced to heart by antiviral therapy, is reduced by the infection, but antiviral therapy can keep life expectancy quite high. I mentioned to you that there are a couple of years less than HIV negative people. Morbidity is a problem with ART because the therapy can reduce the morbidity and can prevent, of course, the transmission of the virus. Because there is still a stigma and discrimination of patients with HIV infection. And, of course, because there are, last but not least, there are possibilities to use the antiviral therapy for many years and have a very high adherence, a relatively low toxicity, but we have to consider the costs of the infection as such. I mean, if you cure the virus, you reduce the long-term costs of the population, of the health system, and especially in countries where we have a public health system, like here in Italy, we pay a huge amount we pay taxes, actually very huge amount of money goes in taxes, but we have a public system, and so the public health, nobody has to pay for the drugs, nobody pays for hospitalization and everything, but the result is that the calculation was that if you do not prevent the infection, and if all patients with do not undergo treatment, the cost all over the world is supposed to be of the order of $22 billion per year to support treatment, prevention, and care of the infection. So it is clear that the optimal result of therapies and strategies would be the eradication. How can you reach this goal? It is not very simple. I mean, eradication means that you can invent or you can create a sterilizing cure in order to eliminate all the HIV-infected cells. You have to go to use methods to reach the viral RNA in plasma below 1 copy per ml. That means really, really undetectable. And there are examples. There is a very nice and very big example is the burning patient, and I will show you in a moment what happened in the case of the patient from Berlin, Germany. The remission is different from eradication. Remission is not a sterilizing cure, it is a functional cure, and the remission means that you can try to reach a long-term health status without any antiviral therapy, reaching the level of HIV RNA in plasma below 50 copies per ml, and there are patients who are able to do this, and these very lucky patients are defined long-term non-progression and actually elite controllers, and post-antiretroviral therapy controllers. These are patients able to control the virus after therapy, and they do not need therapy for some long periods because the immune system that they have is strong enough and able enough to control viremia and virus production. Actually, there is one unique example of a successful cure for HIV infection, which is called the Berlin patient, Timothy Brown. It is a person who had a tumor and leukemia, and he received a transplantation of cells from a donor, allogeneic donor, I mean a person with a completely different HLA, but this patient, this donor, had a mutation in the CCR5 receptor, which is a receptor for a chemokine, and the receptor has a homozygous delta 32 mutation, so this receptor is used from the virus to enter the cell together along with the CD4 molecule. So a cell, the virus, and the HIV uses both the CD4 molecule and the chemokine receptor CCR5 in order to enter into a cell and to infect the cell. If you have a mutation in the CCR5 receptor, it may happen that the virus is not able to enter the cell anymore, and patient, you are in a way protected from the infection. So what happens? That this patient with leukemia receive a bone marrow from a donor who had a mutation in this receptor, and the result is that it was not infectable anymore. So the virus was not present in this patient for a long period. For a long period means that he had HIV infection and leukemia. He was treated for leukemia, and now, after some years, nobody was able to find any sign of the virus in him. So he was considered cured from HIV. Unfortunately, there had to be something more than that in this patient, because the same treatment has been tested in other patients with HIV infection and leukemia. And for example, there are two cases in Boston on two patients who received the same treatment. I mean, that means they received transplantation with the death of cells with the delta-32 mutation, but unfortunately, these are the graphics showing how the virus went to undetectable levels for many years after transplantation. Unfortunately, what happened is that in both on the two patients had relapses and had to go back again to the antiretroviral therapy. So the cure, the sterilizing cure, was not reached in those cases. And actually, there are also other cases described. There is one baby in Mississippi which was very famous a couple of years ago, and another case in Milano in Italy, and also in that case there was a baby who received a therapy immediately after birth, and she was off therapy for many years, but the virus came out again. So it means that sterilizing cure is very likely to be performed. So what about the antiviral therapy and the persistence of the virus? As I told you, we have a number of drugs. Now you can take one single pill per day or a little bit more than that. But if you stop therapy, you see here that when you stop the treatment, in time zero, after a few days, you immediately restart to produce the virus. So it means that even if for many years the virus has been controlled by the therapy, in the moment in which you stop the therapy, the virus can come out again and induce infection of other cells. So to make a very simple scheme, the production of the virus is the phenomenon by which a cell after activation starts to produce virus anymore. But if you put antiretroviral therapy, what happens is that the DNA of the cell is occupied in part by the DNA of the virus. I mean, the virus becomes from RNA, goes to DNA and integrates into the cell and can reach the status of resting for many, many years. So what happens is that the latency of the virus means that the virus is present but not active. So it means that within a cell it is not producing other viral particles. Of course, the virus is able to persist, integrate in the genome of the host, and can be hidden, remain hidden from the immune response, because if no molecule are produced, the virus is not. It is not possible to kill the cells that have the infection. So reservoirs are the places where the virus remains, and they persist even when patients are on antiviral therapy. How can you measure the reservoir? There are many, many systems to measure the presence of infected cells that do not produce virus, and to do this, what you have to perform are some assays activating cells and to see how activated cells produce a virus. But where are the reservoirs? As I mentioned, reservoirs are dispersed throughout the body. It can be in the brain, in the lymphoid tissue, in the bone marrow, in the genital tract. And the nature of the virus is very complex, because you can have a number of cells with different phenotypes that are actually infected by the virus and do not show any sign of the infection until the virus is produced anymore. Another problem is the size of the reservoir, because it varies, it changes with time. time from infection, time of therapy, and so on. So there are many problems related to the measure of the reservoir. And it is clearly important to analyze the reservoir to understand whether a cure, eradication cure, has been achieved, or which is a possible intervention that you can do in order to use agents able to induce eradication. How can you measure the reservoir? As I mentioned, there are many systems. There are methodologies based on cell culture, which are very long to perform and very complicated. There are assays measuring the genes of the virus. And the problem is that many of the assays are not feasible in many resource-limited settings because they can require cold chain logistics, machinery, expensive machinery, and time, and many nations are absolutely not able to perform this. So a large barrier exists in to reproduce these assays and to perform analysis of the reservoir. Many people try to revert the reservoir and the latency of the virus activating cells, and there are systems to perform what has been called prime shot and kill. And the logic is that some laboratories try to activate in patients the reservoir by using vorinostat or other drugs that are able to induce the production of the virus and to show to the immune system that cells are infected and can be killed by CD8 cells. However, the results for the moment are not really easy to understand and to reproduce because of many reasons, one of which is that clinics and laboratory has to be very well organized. The point is that in any case there is a vicious circle because the virus infection provokes immune activation that increases the number of infected cells. Immune activation is linked to inflammation, and inflammation and activation are linked to an incomplete and altered immune recovery. So what can be done is to understand how activation and inflammation are linked to the infection, and we know very well that the activation and inflammation are linked to mortality and morbidity. And many trials showed that the presence of cells, of markers in cells, as well as molecules that are linked to immune activation and to inflammation, are very strongly predictors of morbidity and mortality. And by the way, inflammation is very much linked to chronic immune activation, and chronic immune activation means that you exhaust the immune system, and the immune activation involves a huge number of bacteria, of cells, like those produced in the thymus, those that are responsible for cellular and humoral immunity, and you go and you exhaust the reservoir of the immune system. We know that immune activation is very dangerous for the organism. From the animal models that the group of widows in Leicester set up many years ago, we know that the more the immune system is activated, the more you produce virus. And the more you produce virus, the more you kill immune cells, including precursors. In the immune system, precursors are in the thymus, and you can measure the activity of what happens in the thymus with different methods. So the best method is to measure the TREC cells, TREC positive cells, that are cells that have the T cell receptor rearrangement circles in the cytoplasm. We can measure easily TRECs with different methods, and I will show in a moment how we can do this. It is very well known that the TREC are linked to the THYMIC volume, and the THYMIC volume is important as a marker to understand how the immune system is destroyed or is altered by the virus. TREC, thymus is important not only in children but also in adults. And the volume of the thymus, that means the functionality of the thymus, is crucial in order to have a good immune reconstitution. What can be done is to measure TRECs. TREC is the T cell receptor rearrangement excision circles, which are what happens during the rearrangement of the cells. To make a long story short, what happens is that during the rearrangement of the alpha chain of the T cell receptor, some parts of the DNA are cut. They form circles that remain in the cytoplasm and can be measured by molecular biology essays. These circles dilute when there is a proliferation of thymocytes, and they are diluted during clonal expansion. So at the end of the day, if you measure the amount of TRECs in blood of cells that are TREC, you have an idea of the age of the thymus and of the activation on the immune system. And many studies have been performed showing that the changes in the functionality of the thymus is linked not only to the age of the patients, as it happens, but also to the state of HIV infection, and that the number of TREC cells decreases significantly with the infection. Some years ago, we showed that patients who have a very high immune reconstitution were also able not only to maintain the integrity of the thymus, but also to have a high amount of TRECs in plasma, in the blood, cells which express TRECs. And these data were confirmed many years later by a group in France. In 2014, it was shown that the antiretroviral therapy, the treatment, can increase the amount of TREC-positive cells in the blood. So it means that measuring TREC is important to understand how the immune reconstitution occurs in patients. On the other hand, we know that persistent inflammation is crucial, that persistent inflammation induces a chronic activation, and a chronic activation destroys the immune system. So measuring parameters which are related to the immune system means that we can understand not only what happened to the side of the virus, because the virus is very well controlled by therapies, but also on the side of the immune system. And which are the strategies that we are using to measure the activity of the virus and the activity of the immune system? Well, we have set up different technologies. One, to evaluate the HIV proviral DNA by digital drop PCR in highly purified sorted cells. We have also measured the presence of THYMIC output by measuring TRECs. And finally, we also measure the age of immune cells by analyzing the telomere length. The system that we use now, as I mentioned before, is a cell sorter, which is a S3 instrument from Bio-Rad, of course, and a cabinet. This is a cabinet biobubble that allows you to work with infected cells without any risk for the operator. The machine, this instrument, is crucial to sort cells and to perform the study that I am going to present you, in which we have analyzed 32 patients with HIV infection, which were successfully treated for many years, and the mean age was 49 years, 11 females. For this count was over 500 cells per microliter, and viremia was undetectable for at least 1 year. So it means that they did not present the activity of the virus in the plasma anymore. These are the characteristics. With the time on therapy is the mean is more than 11 years. The viremia in the mean undetectability was 72 months, and the count before therapy was 329 cells per microliter, after therapy 700 cells per microliter. So it means that the therapy not only killed the virus, but also was able to induce an increase in the number of CD4 cells. What we have done first was to sort different populations of T lymphocytes, of CD4 cells. So we measured the amount, we sorted naive cells, we sorted central memory and effector memory cells in order to understand whether they present different amounts of the virus. This is the strategy of sorting. We could sort at least 300,000 cells per subset, naive, central memory, and effector memory, with a S3 sorter, with a purity of higher than 95%. Of course, we had all the controls. And what came out is that the distribution of naive, central memory, and effector memory and terminally differentiating cells was different among different populations. As we know, patients have a high amount of naive cells because of the therapy. About 50% of CD4 cells were naive, about 25 were central memory, 20 were effector memory, and very few of them, less than 5%, were terminally differentiated. So what we could do was to use a system by the new CFX96 Touch real-time PCR instrument that was coupled with a QE200 digital drop PCR system that we have in our lab to measure the amount of HIV present in the different CD4 cell population. Actually, the methodologies were very well known because there are several groups that have developed the technology to measure HIV DNA within cells, and these are some examples of what we have done with the quantification of the proviral DNA in terms of LTR by digital drop PCR. This is the quantification of the virus in the two channels. So we measure on the one channel, on channel 1, the LTR from the virus, in channel 2, the eukaryotic translation initiation factor 2C1, which is a very well-known gene, a reporter gene present in all cells. And what came out is that, well, these are just some graphics to show you how the system works and how the system is very sensitive, being able to detect very, very few amounts of infected cells per sample. And what came out is that DNA content was lower in naive cells than in central memory or in effector memory, which was expected. So we already knew this from the literature, but it is always nice to confirm some data. And so in naive cells, the amount of virus was about one-third of what happens in central memory or in effector memory cells. What came out is that the analysis of our data showed that the amount of CD8 cells is very much related to the amount of virus. In fact, if you divide patients in those who had more than one or less than one copies of LTR per cell, well, those that had low amount of copies of virus had more CD8 T cells. So it means that the amount of CD8 is crucial to control viremia, intracellular DNA. Actually, we saw that the nadir of DNA of CD4 cells did not affect the content of HIV DNA. So it means that nadir is the lowest number of CD4 cells during the story of the patient. So it means that if you had a low amount of CD4 before therapy, or if you had a high amount of CD4 cells before therapy, you can still become undetectable and you have the same amount of the virus in your blood after therapy. So it means that patients who had a nadir below 314 cells per microliter, or those who had a nadir higher than this value, had the same amount of the virus in the central memory, effector memory, or naive CD4 cells. So the amount of DNA is not affected by the nadir, but it is very much affected by the time of therapy. So patients who are on therapy for more than 10 years have a significant amount of virus lower than patients with therapy that started before that period. So it means that the length of the treatment is very important in order to control the virus within cell. So viral DNA is lower in patients with longer time of treatment. So it means that treatment has to be started as soon as possible. Similarly, another interesting data that came out is that the ratio CD4-CD8 is very important because those patients who had a higher ratio CD4-CD8 had a lower viral content into naive cells as well in central memory and in effector memory. So it means that the ratio CD4-CD8 is very much indicative of what happened in the viral compartment. Then we took a look to the immune system, not only to the virus, but we quantified cells positive to TREC and the thermal length to evaluate cell senescence. The strategy to measure the TREC were published some years ago, and what came out is that, again, we could quantify the amount of TREC in naive central memory or effector memory, and what came out is that, again, the methodology is very nice, it's very precise, the method is very simple, and you can see in the graphic that it is very clear the presence of cells that had TREC or that had either TREC and the reported gene is very easy to analyze. Well, what came out is that TREC was very much present in naive cells as expected, and at very low level was present in central memory or effector memory. But interestingly, we found some strange correlations between the amount of TREC and the viral load. So it came out that changes in the viral load in amount of virus were not related to the presence of TREC in effector memory or in naive cells, but there was a correlation between the amount of cells that express TREC and the viral presence within such cells. Actually, terminal length had no difference. We did not, I not show you graphics concerning different populations of patients because we found actually no changes in the terminal length in all patients and in any other system we try to analyze. So in conclusion, I hope that I could show you that the combination of flow cytometry and cell sorting with molecular biology and approach based upon digital PCR can give precious information on the amount of the virus present in different CD4 T-cell populations. We showed that you can measure the capability of the immune system to regenerate, and maybe this could provide a guidance for the monitoring of patients with HIV infection. And of course, finally, we think that the combined approach could lead not only to a better comprehension of the reservoir, because again, all the analyses were done in patients with undetectable viral load. It means that no virus was present in plasma. What we have done was only in cells taken from the blood. So we sorted different populations of CD4 cells, and we saw that in different populations of CD4 cells, there were different amounts of HIV DNA. So infection was present in the cells, in the blood, and could be measured. Of course, this could be important, useful to develop different strategies of eradication of the infection. And finally, I would like to acknowledge my collaborators. First of all, Professor Cristina Mussini, the blonde lady on the left, who is my wife, by the way, and she's the director of the infectious disease clinics. Professor Marcello Pinti, who is a very clever molecular biologist, and we have been working together for many, many years, and all with Sara, Lara, Milena, and all the others. And of course, I want to thank you for your attention, and I'm here to take any questions.
Thank you, Andrea, for that interesting presentation. Now let's move on to the question and answer session. If you have any more questions, you can submit these at any time during the webinar. The first question says, what was your experience using the S3E cell sorter, and was there a particular reason that you chose that instrument?
Well, the experience we had is actually now it's very, very good. We had the first instrument here in my lab for some time. We worked on that. We set up all different methodologies, and the reason for the use of the instrument is that, well, it is very simple. It's very simple. It's all many things are automatic, like the alignment on the laser, the quality control of the samples, and everything. And there is the possibility to have remote control, so if there is a trouble somewhere, someone from the US, from UK, from Holland, I don't know from where, they can fix the problem in a remote mode. The instrument can go under a cabinet, which is very important, because in this way we can sort cells in a sterile environment on the one side, and on the other side in a way that it is completely and absolutely safe for the operator. And this is very important because I see many times in different labs all over the world people who do sorting without respecting the rules of a good sorting practice. So it means that you have to avoid the presence of aerosol and stuff like that. So you need to put the instrument under cabinet. And the sorter we have is very, very good for this because it is not so big. You can put under the cabinet and work perfectly. And another good thing is the software, which is easy, and the sensitivity and specificity. So we can sort with a purity of 99%, actually with very no problem at all. And many people do this in my lab, so we don't need to train people with complicated instruments because it works well, and it's very easy. And actually, I'm not aware of the price that the instrument has everywhere, but the price is more than reasonable, so it is not that I have to sell my house to buy a cell sorter.
Thank you, Andrea. Our next question asks, you have created a nice method combining the sorting of T cells with digital PCR. What experiments are you planning to do next?
Well, in this moment, we have developed also another system to measure different forms of the virus. So in a short time, we will have the analysis of the integration of HIV in cells and the analysis of the circular forms of the virus. This will be actually is ongoing, so I think that we will have the data in a few weeks on the side of HIV. But actually, we are moving our interest in another field, which is of great importance in the therapy, not for HIV infection, but for cancer. So we are developing now strategies to study cells in patients who are treated with new biological drugs for cancer, which are the anti-PD-1 and PD-L1, the new compounds that are able to inhibit the inhibitor checkpoints of the immune system. And these are new assays that we are developing to measure the activation of cells during treatment and to find possible biomarkers which are related to the capacity of the patient to respond to the therapy or not, which is a crucial issue actually in the field of cancer therapy.
Thank you for that. Our next question asks, what are your thoughts on using gene editing in immune cells to reduce the rate of HIV infection?
I'm sorry, can you repeat? I cannot hear well.
You discussed several approaches for HIV cures. What are your thoughts on using gene editing in immune cells?
That would be absolutely important. Of course, gene editing is crucial. The question is, the point I always raise when I discuss with my colleagues is that all approaches have to be feasible on a large scale. So you have to divide the question in two parts. What could be done and what can be done? In theory, if you do gene editing, if you have the zinc finger proteins and you can really manipulate cells, have gene expression as you want, edit some genes and remove parts of the genome, but this is not, and this can be done actually in laboratory, in tubes, not on patients. It's not feasible on patients, first. Second, the costs are absolutely not affordable, so it cannot be even thought on a large scale. On a large scale, what could be designed, what the goal, the holy grail of HIV infection treatment is, of course, a vaccine. A preventive vaccine to block the spreading of the infection on the one side, a therapeutic vaccine to help the immune system to remove and to kill infected cells. So these are the two goals that we have. But in my opinion, also performing strategies of gene editing and killing cells by having them recognized by the immune system is a very nice proof of concept because people have to show how you can kill the virus. It's a proof of concept. I don't see this as a feasible therapy, but it has to be done to understand even better the response of, the possible new, and to understand which are the streams to follow to have new strategies for fighting HIV infection.
Thank you very much. That's all we have time for today. Thank you to Andrea for today's informative discussion and presentation, and also thank you to everyone joining us online and for your interesting questions. I hope that you found this a worthwhile session. If we didn't manage to answer your questions, please feel free to email me at lindsay.forsyth@selectscience.net, and I will follow up on your questions later. If you would like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand very shortly. Goodbye, and thank you once again for joining us.